This method can help answer key questions in the Dictyostelium and Macropinocytosis fields, such as whether a particular gene, protein, or molecular pathway is involved in macropinocytosis. The main advantage of this technique is that many variables such as time, genetic mutations, inhibitor concentrations, and media compositions can be tested in parallel, giving results with single-cell resolution.
To begin this procedure, obtain cultivated cells on SM agar with bacteria as outlined in the text protocol. Harvest cells from the feeding front into 25 milliliters of KK2 buffer in a 50 milliliter centrifuge tube. Vortex the tube to dissociate the cells. And centrifuge at 300 G for 3 minutes. Next, discard the supernatant and resuspend the pellet in 50 milliliters of fresh KK2 buffer. Repeat this wash 3 times by centrifuging, removing the supernatant, and resuspending in 50 milliliters of KK2 buffer for each wash. After this, use a hemocytometer or other cell-counting device to determine the cell density. Dilute the cells into HL5 medium containing antibiotics to a concentration of 100,000 cells per milliliter. Pipette 50 microliters of the diluted cell solution into each well of a flat-bottomed 96-well tissue culture plate, using 3 wells for each condition. Incubate at 22 degrees Celsius for 24 hours. Then, dilute trizidextran to 1 mg per milliliter in the medium used. Add 50 microliters to each well for a final trizidextran concentration.